Yes, good morning, all of you again. Chaliye. So, last session we understood a beautiful topic called as the dark reaction, and today we have a very nice logic called as the chemiosmotic hypothesis. Sounds familiar? In mitochondria, ATPs were made by the chemiosmotic hypothesis. If you remember, during the ETS, hydrogen and electron liberate energy, and that energy took proton from the matrix into the intermembranous space, and there was a high concentration of proton in the intermembranous space which returned back into the matrix along the proton gradient, along the concentration gradient, which helped in the formation of ATPs in the F1 particles with the help of ATP synthetase or ATP synthase enzyme. Similarly, in chloroplast, we talked about it, cyclic and non-cyclic photophosphorylation produce ATPs. In cyclic photophosphorylation, two ATPs are made. In non-cyclic photophosphorylation, one ATP is made. And if you remember, cyclic में the two ATPs were made. If you remember, cyclic में the two ATPs were made between ferredoxine and cytochrome B6 and cytochrome B6 and cytochrome F. And in non-cyclic, the two ATPs were made, or rather the ATP was made, one ATP was made between cytochrome B6 and cytochrome F. यहाँ ATP बनता है. Actually, these ATPs are not made between ferredoxine and cytochrome B6 or cytochrome B6 and cytochrome F. Actually, these ATPs are made by a similar mechanism called as chemiosmosis. By chemiosmotic hypothesis, ATPs are made. तो फेरिडॉक्सिन और साइटोक्रोम बी सिक्स साइटोक्रोम एफ, इनके बीच में सिर्फ इलेक्ट्रॉन से एनर्जी जाती है, and that will help in movement of proton. What is the difference? So the difference is in case of respiration. Sir, ये था आपका माइटोकॉन्ड्रिया and ये थे आपके क्रिस्टे, and protons went from the matrix into the intermembranous space. And from intermembranous space, they returned back through the F1 particles. And F1 particle में ATP synthase trapped the energy to form ATPs. तो ये mitochondria में था. Where did protons return back? Protons returned back from the intermembranous space back into the matrix, और उसके अंदर F1 पार्टिकल के अंदर ATP बनता है. A similar chemiosmosis is seen where we have the chloroplast, और chloroplast के अंदर here we have grana, grana के अंदर here we have thylakoid, and thylakoid के अंदर here we have the lumen of the thylakoid. Lumen of thylakoid. And lumen of thylakoid, and surrounding the lumen of the thylakoid is the stroma. तो ये है stroma, और stroma के बीच में ये है lumen of the thylakoid. उनके बीच में होता है chemiosmotic hypothesis. So, where was chemiosmosis in respiration? between the intermembranous space and the matrix. Where is chemiosmosis in photosynthesis? Between the stroma and the lumen of the thylakoid. Beautiful process. Let's understand this. Third, in case of chloroplast, here we have chloroplast और chloroplast में ये है stroma, ये है stroma and stroma के अंदर ये है grana and grana is made up of units which are called as thylakoids. तो ये part है stroma और ये है grana and grana के individual units are called as thylakoids. Inside the thylakoid is the lumen of the thylakoid. तो thylakoid के अंदर ये है lumen of the thylakoid. The stroma is rich in H plus ions. Stroma में बहुत सारे protons होते हैं. Stroma has lot of protons. And third, ध्यान से सुनना. I hope you remember, we have photosystem 2, PS2 or P680 on the inner membrane of the thylakoid. याद है मैंने बताया था. And on the outer membrane of the thylakoid, we have photosystem 1 or P700. And we have NADP. If you remember, in non-cyclic photophosphorylation, electron goes from PS2 to PS1 and PS1 से electron goes to Z, then to ferridoxine, and from ferridoxine it goes to NADP, and NADP is present on the inner membrane of the thylakoid. So PS2 is present on inner membrane, PS1 is found on outer membrane, and NADP, nicotinamide adenine dinucleotide phosphate, is present on the inner membrane. And you have NADP gets reduced to become NADPH2. NADP gets reduced to form NADPH2 with the help of an enzyme called as Nadp reductase. Do we remember in case of respiration, we had a similar enzyme called as Nad reductase. यहाँ पर है NADP reductase. So NADP reductase enzyme, there is this enzyme found on outer membrane. So once again, PS2 is found on inner membrane, PS1 is found on outer membrane, and NADP is found on inner membrane, and NADP reductase is found on outer membrane. This is a beautiful structure of the thylakoid. And here we have thylakoid ka peristromium. Peristromium has in it particles called F1 particle. In case of mitochondria, we had base, we had stalk, and we had head. 3 parts. In case of chloroplast, we have something called F0, which is the transmembranous channel. तो वो membranous और transmembranous channel होता है, which is found in the peristromium. Periplastidial space में होता है. And you have F1 particle which is found in the stroma. F1 particle is outside the thylakoid in the stroma. And F1 particle, so ये है F0 और ये है F1. F1 is found in the stroma and F1 particle has in it the same enzyme. If you remember, the enzyme called ATP synthase enzyme. So ATP synthase enzyme is found in the F1 particle. Now, what will happen during cyclic and non-cyclic photophosphorylation? Cyclic and non-cyclic photophosphorylation में PS2 and PS1 receive photons, and PS2 at 680 nanometer wavelength loses its electron. PS1 at 700 nanometer wavelength loses its electron. तो समझो नॉन साइक्लिक प्रोसेस हो रहा है. Who will lose electron first? If you remember, PS2 at 680 will lose electron first. तो सबसे पहले PS2 का इलेक्ट्रॉन शूट आउट होगा. Who will accept the electron? If you remember, quinone, and from quinone it will go to plastoquinone. From plastoquinone to I hope you remember, cytochrome B6, cytochrome F, plastocyanine, and electron reaches plastocyanine. In the meantime, PS1 reaches 700 nanometer wavelength and loses its electron, and electron goes to Z, and from there to ferridoxine. And so, where is the electron found? PS2 का electron is with plastocyanine, PS1 का electron is with ferridoxine. And from plastocyanine, the electron goes to PS1 and brings PS1 back to a ground state. PS1 ground state पे आ जाएगा, and from ferridoxine, the electron goes to NADP, and NADP absorbs or retains the electron. तो PS2 का इलेक्ट्रॉन PS1 के पास चला जाता है, and PS1 का इलेक्ट्रॉन NADP के पास चला जाता है. Now, how will so who brought PS1 back to ground state? Electron from PS2 brings PS1 back to ground state, and PS1 का electron has gone to NADP. Who brings PS2 back to a ground state? I hope you remember. There will be photolysis of water, and water molecule breaks to form proton and rather H plus and OH minus. So H2O splits to form H plus and OH minus, of which OH minus forms electron and OH, and electron goes to PS2 and brings PS2 back to a ground state. So where is the flow of electron? Flow of electron is from PS2 to PS1 and PS1 to NADP, and from water electron goes to PS2 and brings PS2 back to a ground state. Chalo, so H2O has split to form H plus and OH minus. OH minus का electron has gone to PS2 and brought PS2 back to a ground state. But where has this photolysis of water taken place? Photolysis takes place in the lumen of the thylakoid, क्योंकि PS2 was found on the inner membrane. तो PS2 inner membrane में है, so photolysis takes place on the lumen of the thylakoid. And what does H2O split to form? H plus and OH minus. So OH minus ka electron goes to PS2, brings it to ground state, but there is accumulation of H plus. So proton accumulates inside the lumen of the thylakoid. Proton accumulates into lumen of thylakoid. So protons move into the lumen of the thylakoid because of photolysis of water. So photolysis of water makes proton accumulate in the lumen of thylakoid. Photolysis of water make proton accumulate in the lumen of the thylakoid. And what else will happen? Sir, PS2 का इलेक्ट्रॉन तो PS1 के पास चला गया, PS1 का इलेक्ट्रॉन NADP के पास चला गया, and NADP retains the electron. NADP retains the electron, so it will become negatively charged. NADP with electron becomes negatively charged. And if you remember, stroma is rich in protons. So H plus proton from the stroma will now have an electrostatic attraction for negatively charged NADP minus, and plus moves towards minus. So protons move from the stroma through the thylakoid towards NADP minus, and NADP minus के पास protons जाते हैं, and protons from the stroma start moving into the thylakoid. तो thylakoid के lumen में protons accumulate होते हैं because of NADP minus minus. If you remember, one NADP can attract or absorb 2 electrons. One NADP can have 2 electrons, which then combines with 2 proton to form NADPH2. So NADP minus minus now has a strong affinity for protons, and protons move from the stroma into the lumen of the thylakoid. So lumen of thylakoid में protons go from the stroma again into the lumen of the thylakoid, and lumen of thylakoid में proton accumulate हो जाएगा. So protons have moved into the lumen of the thylakoid now by 2 reasons. First reason we remember was photolysis of water, and second reason is NADP minus minus. So photolysis of water made H plus accumulate in the lumen of the thylakoid, and NADP minus minus also made proton go from the stroma into the lumen of the thylakoid. तो photolysis से भी H plus आया, and NADP minus minus का electrostatic force of attraction से भी proton आया from the stroma into the lumen of the thylakoid. And what else, sir? Sir, do we remember NADP minus minus has to become NADPH2? NADP minus minus has to become NADPH2. But for that, you need presence of an enzyme. And that enzyme is called as NADP reductase. NADP reductase will combine with NADP minus minus with proton to become NADPH2. But beautiful, where is this enzyme NADP reductase found? On the outer membrane. सो NADP रिडक्टेज का काम है प्रोटॉन को लेके NADP माइनस माइनस के साथ कंबाइन करके NADPH2 बनाने का, but NADP reductase is found on the outer membrane. So because it is found on outer membrane, it will now start picking up protons from the stroma, and from the stroma, protons will be pumped from the stroma again towards NADP minus minus, which means protons will accumulate inside the lumen of the thylakoid. तो ये वापस से प्रोटॉन ल्यूमेन ऑफ थाइलाकॉइड में आ जाएगा. अरे which means अब 3 रीजन हो गए. There are 3 reasons why protons move into the lumen of the thylakoid. Which was the first reason? Photolysis of water. H2O split हुआ to form H plus and OH minus. तो उससे H plus accumulate in the lumen of the thylakoid. Secondly, NADP minus minus, which has received electron from PS1, NADP minus minus have an electrostatic force of attraction for proton, which will go from stroma again into the intermembranous space. And third reason, to NADP minus minus. And third reason, NADP reductase is found on the outer membrane. It will bring proton from the stroma into the lumen of the thylakoid, so it can combine with NADP minus minus and form NADPH2. तो 3 reasons से look at this proton concentration. Proton concentration in the lumen of the thylakoid will tremendously increase. So once again, what are the 3 reasons why protons move into the lumen of the thylakoid from stroma? First, we had photolysis of water. Photolysis of water. Secondly, we had NADP minus minus having an electrostatic force of attraction for protons from the stroma. And we had third reason, NADP reductase found on the outer membrane, which will bring protons from stroma towards NADP minus minus to combine with it to form NADPH2. And ये 3 reason से protons accumulate in the lumen. याद रखना, I've written it specifically. 3 reasons. Photolysis of water accumulates proton. NADP minus minus attract proton. And NADP reductase bring in proton. And proton concentration inside the lumen of the thylakoid will increase. Hey, sounds familiar. Sir, in case of respiration, proton concentration in the intermembranous space had increased. याद है? Similarly, in photosynthesis, proton concentration in the lumen of the thylakoid will increase. And in respiration, where did protons go? From the intermembranous space where the concentration was high, they moved back into the matrix along the concentration gradient. That was called proton channel pumping. Similarly, in photosynthesis, where will these protons go? These protons have gone from the stroma into the lumen. From stroma, they have gone in the lumen. So, lumen में proton concentration has increased and stroma में proton concentration has decreased. So, where will protons go? Back from the lumen into the stroma. So protons will go back from the lumen into the stroma. So protons move from high concentration back into low concentration. From high concentration back into low concentration. क्योंकि स्ट्रोमा में प्रोटॉन का concentration हाई था, but for all these three reasons, protons have moved into the lumen of the thylakoid. And in the thylakoid, in the lumen of the thylakoid, concentration of protons has increased. So protons will return from high concentration back to low concentration along the concentration gradient. Along the concentration gradient, which is also called protons move from lumen of thylakoid into stroma along the proton gradient. So what is that gradient called as? Proton gradient, which is along the concentration gradient. And as protons pass from the lumen into the stroma, from lumen back into the stroma, where will they pass? If you remember, the entire intermembranous space has in it F0, F1, F0, F1. तो ये है आपका F0 और ये है आपका F1 and F0 is the transmembranous channel in the peristromial space. So protons pass through F0, F1, F0, F1, F0, F1, F0, F1 and as protons pass through F0, F1 everywhere, F0, F1, F0, F1 and as protons pass through F0, F1, what will happen? As a proton passes through F0 into the F1 particle and returns back from the lumen into the stroma, it will pass through F0, which is the transmembranous channel, then through F1 particle. F1 particle has in it ATP synthase and ATP synthase will trap that energy due to movement of proton. The kinetic energy of movement of proton is trapped by ATP synthase and combined with ADP and IP to form a molecule of ATP. So again, what is this phenomenon? This is formation of ATP due to movement of proton from the lumen into the stroma along the proton gradient. So high concentration, say low concentration along the proton gradient, movement of proton from lumen into stroma makes proton pass through F0 and then into F1 particle or F1 particle may we have ATP synthase and ATP synthase combines with ADP and IP to form a molecule of ATP. What is this phenomenon called as? Chemiosmotic hypothesis. इसे क्या कहते हैं? Chemiosmotic hypothesis. Beautiful, lovely process. So I hope you've understood. याद ये रखना है एटीपी बनता कौन से रीजन से है? Protons accumulate in the lumen due to three reasons: due to photolysis of water, due to NADP minus minus, which gets electron from PS1 having an electrostatic force of attraction for proton, and NADP reductase bringing proton from stroma back into the lumen of the thylakoid. So NADP minus minus can combine with proton to form NADPH2. तो तीन रीजन से प्रोटॉन ल्यूमेन में आ जाते हैं, तीन रीजन से प्रोटॉन ल्यूमेन में आ जाते हैं, that is photolysis of water, NADP minus minus and NADP reductase. And as proton concentration in the lumen will increase, the protons will move from high concentration back into low concentration. Protons will move from high concentration back into low concentration along the proton gradient or concentration gradient. And as protons go from the lumen back into the stroma, they had gone from stroma into lumen, and they have gone from lumen back into stroma along the proton gradient, along the proton gradient. And while passing through proton gradient, they pass through F0, F1. F1 has in it ATP synthase, which combine with energy, kinetic energy of the movement of proton, and combine it with ADP and IP to form a molecule of ATP. Beautiful. What is this phenomenon called as chemiosmotic hypothesis? Excellent logic. I hope you've understood this. Beautiful point. Chalo, इसे याद रखना. We will now analyze, or we have analyzed, the concept of chemiosmosis.